Hello, assuming you now know a bit about aptamers, I'm going to talk to you about SELEX, what it is, and what you're going to be doing in the lab. What is SELEX? SELEX is an acronym for Systematic Evolution of Ligands Through Exponential Enrichment. I know it's kind of a mouthful, but in simple terms, SELEX is the process used to find aptamers. It takes multiple steps to complete a round of SELEX, and it takes multiple rounds of SELEX to find an aptamer, but we'll talk more about that later. As a quick reminder, an aptamer is a short sequence of single-stranded DNA or RNA that has an affinity for a target molecule. The relationship between SELEX and aptamers is that SELEX is the process and the aptamer is the product. In other words, you do SELEX to get aptamers. Since SELEX was discovered in the 90s, researchers have developed many different variants. However, today, we are just going to focus on the type of SELEX that we'll be doing, capture SELEX. For us, SELEX begins with an initial library of single-stranded DNA sequences that contain a random region in the middle of the strand with fixed constant sequences on either end. This random region is usually between 30 to 80 nucleotides long. With all the different possible arrangements of the A, T, C, and G base pairs, we estimate that our library, with a random region of 40 nucleotides, has about 10 to the 14th unique single-stranded DNA sequences. Ideally, at least one of these sequences binds to our target molecule. I would say that SELEX is like searching for a needle in a haystack, where the haystack is this massive pool of short and random DNA sequences, potential aptamers, and the needle is the aptamer, the short and random DNA sequence with the capacity to bind to a desired target molecule. When we start this process, we have no clue which DNA sequences might have an affinity to the target molecule. Our way of addressing that is by using a random pool of DNA so large and so diverse that it becomes statistically probable that an aptamer will appear. We're sifting through 100 trillion different possible candidates at once. If that's the explanation for the random region, what are these constant sequences on the side? These constant sequences are a really important tool because we know how the constant regions will behave. This will become more apparent as I explain the first part of SELEX, positive selection. This part is how we determine which of the potential aptamers actually have an affinity for our target molecule. The first step is just to mix the initial random library with a biotinylated capture strand. In this diagram, I've picked a couple of strands with different colors to represent our random library. The biotinylated capture strand is just a short complementary piece of DNA. It is biotinylated because it is tagged with biotin. That's just a fancy way of saying that. This part is the biotin and this part is the complementary DNA. Biotin is a small, stable molecule, and we'll see what it's used for in just a second. The complementary DNA on the capture strand will bind to the library, creating a capture library hybrid. Every potential aptamer in the library has the same constant region, so it is really easy to generate this complementary strand. The second step is to take a flow column and fill it with streptavidin-coated beads. The capture library hybrid can then be added to the column as well. Streptavidin binds tightly to biotin, so the beads will be able to hold all the capture library hybrid in place. At this point, you could say that the capture library hybrid is immobilized. Let's take a closer look at the connections that are happening here. The biotin holding to the streptavidin is a really strong bond. Once they're together, they won't really want to let go. The bond between the two strands of DNA, like the capture and the library, is much weaker. Now that everything is set up, the target compound can be added to the column as well. Here is a diagram of everything without the target compound, and then here is the target compound added in. This is the part that is truly positive selection. The DNA strands with an affinity for the target compound will bind to the target and come off the column, while the strands with no affinity will do nothing. Here we have just one more diagram of the same thing happening. When a potential aptamer is positively selected, the entire strand of library comes off to bind to the target molecule. The capture strand is left bound to the streptavidin in the column. The column is used to separate these good potential aptamers from other strands that might not have as much of an affinity. Otherwise, it would be really hard to physically separate the two. After positive selection, you now have a pool of single-stranded DNA molecules with an affinity for your target compound, potential aptamers. Let's go over everything we've done so far. First, we start with our random library. Then we introduced a capture strand to get our library capture hybrid. Afterwards, we immobilize everything on a column with streptavidin beads. Then we add our target compound. everything with an affinity for the target compound comes off the column, while everything without sufficient affinity is left behind. Okay, so what's next? You have this solution, and it has a lot of good potential aptamers, but then what do you do? In the first round, remember that the initial library is completely random, so there's basically only one of each sequence. What we want to do next is called amplification. This quickly and efficiently replicates a DNA, so there's many copies of each sequence, with the potential to be an aptamer. If we don't do this, it becomes incredibly easy to just lose an important sequence. Unfortunately, PCR creates double-stranded DNA, so you will have to do a few procedures to make your DNA single-stranded again. That's called regeneration, as well as clean it up, which is purification and concentration. The main regeneration step is a lambda exonuclease digest, and the cleanup steps are a phenylchloroform extraction and ethanol precipitation. I'm not going to go over the technical aspects of these procedures right now, as you'll go over them more in depth in the lab. After cleanup, congrats! You've completed a single round of SELEX. Now we have a pool of DNA with much better potential aptamers, as all of these strands of DNA have demonstrated at least some affinity for our target molecule. This then becomes the starting pool for the next round of SELEX. This is a diagram of the whole process, and in order to develop an aptamer, you have to run through this process 10 to 20 times. Each round of SELEX, you lower the amount of target compound you use each time during aptamer selection. This makes the aptamer pool much more specific each round, until you're ideally left with only a couple of highly selective sequences. Going back to the needle in a haystack idea. The general idea is that you're making the haystack smaller and smaller with each round of SELEX, until you find your perfect aptamer. I think that's about all. We've covered a lot of information today, so feel free to rewatch the video, rewind to important points, or ask your professors or TAs if there's anything you'd like to clarify. These concepts might take some time to sink in. There's a lot of them, but we're here to help and support you along the way. Thank you for watching.